especially Padala Tirupati, of this useful and purposeful program, useful to the students who are aspiring for still higher studies in plant sciences. I am also happy to note that 3 very old prestigious colleges, their botany departments have come together to organize this program for the benefit of wider spectrum of students. I am very thankful for inviting me to deliver the keynote address. I am very happy to share a platform with you in this occasion. A few days back, I had the privilege of inaugurating such program launched by GDC Siddipet Autonomous College. As you are aware, botany is an important subject not only in life sciences, but also in all sciences. It provides a wonderful and it is a perennial subject. It has wide applications in day-to-day life and a great scope building the academic career of the students. This course, though specifically meant for PG Botany entrance examination, it is also useful for various competitive examination, just like groups of TSAPC and UPSC also, forest services like that. The knowledge gained through this program is also useful to get seats in the allied subjects like genetics, MSc Genetics, MSc Microbiology, Biotechnology, Bioinformatics, Biochemistry, and many other courses within our state and also in other states of the country. PG studies have become an important step in today's education. After PG, many opportunities are available for the students like teaching, research in the fields of agriculture, horticulture, food industry, etc. And one becomes confident about starting his own self-employment programs also. By and large, this is a wonderful program. I congratulate all people involved in this program and providing an ideal platform to learn that too at free of cost. I hope the organizers have chalked out a very good schedule for 65 days, including feedback. Congratulations to students. I appeal to students to make use of this program very effectively. Resource persons are very qualified and experienced, drawn from all the colleges and universities. I am confident that you will get benefit and get a seat in MSc PG courses. About 1,000 seats are available in our state MSc Botany. Securing a seat is not a great thing, but aim it to get a good rank. If you get a good rank below 100, you are sure to get seat in university campuses, best colleges, where good teaching faculty and lab facilities will be available. I once again appreciate the efforts of Dr. P. Tirupati, the convener and the nucleus of this program, who is the main architect of this purposeful program, and all other, including the principals, HODs who have encouraged and support this program. My best wishes to the student participants. I once again thank for giving me nice opportunity to share and to become one of the participants in this program. Now coming to this keynote address, but just for a while I thought what topic I should take, because botany is a very big subject, right from the viruses, bacteria to biotechnology. And within the given short time, maybe 30 minutes or 40 minutes. Then ultimately I zeroed on a topic, a very important topic, that is photosynthesis. Even photosynthesis is also a very big topic. And normally In a regular class of BSc, MSc, we take about 10 classes, 10 periods to complete the photosynthesis topic. Anyhow, I try to give a glimpse of the photosynthesis right from the historical aspects to the photorespiration importance of the photosynthesis. This is a very good saying about the photosynthesis. Plants perform the photosynthesis. It is a complicated process. Without plants, we would all will be dead, said by Pamela Ronald. No, this is the long and short of the photosynthesis. What happens during the photosynthesis? Photosynthesis takes place in green leaves. This is the platform for the photosynthesis. During photosynthesis, two things are taken. One is the light from sun, another carbon dioxide. The water is taken through the roots from the soil. These three components, light, carbon dioxide, and water, in the platform of chlorophyll or green tissue, they are converted to glucose. And during this process, oxygen is released. So the two things are light, carbon dioxide, and the end products are oxygen and glucose. Photosynthesis can be singled out as a phenomenon which is responsible for life on the earth. It not only provides the food for all the living organisms, it also provides another essential thing, that is oxygen. Without oxygen, we cannot live. Without food, we may live for few hours, few days, but without oxygen, we cannot live more than few minutes. Definition of photosynthesis. We know all of this, what is photosynthesis. Here, the important event that happens is light energy is converted to chemical energy. The energy present in the sunlight is converted to the energy present in the glucose, carbohydrates. What happens during photosynthesis? Light energy is captured and used to convert water and carbon dioxide and minerals into oxygen and energy-rich organic compounds. So this is all not necessary. Now, this is important. What is photosynthesis? Photosynthesis is an anabolic process, energy consuming process. The energy comes from the sunlight. We know metabolism, there are two processes, anabolism, catabolism. Photosynthesis is anabolism, energy is conserved. Respiration is catabolic process, energy is released. It is an endergonic process, energy utilizing process, energy absorbing process. Here what is happening, for example, if you take carbon dioxide is inorganic, energy less or energy poor. It is converted to an energy-rich organic compound, that is glucose molecule, a carbohydrate. It is a reductive process. Respiration is an oxidative process. Photosynthesis is reductive. These are all historical aspects. The most important contribution came from there is Joseph Priestley. He conducted some classical experiments to show that oxygen is released during the photosynthesis, carbon dioxide is consumed. The Priestley classical experiments. Antony Leeuwenhoek, this is Van Ingenhousz, who is often referred as the father of photosynthesis. Then there are so many people who have contributed for our understanding of the photosynthesis. Then Van Moll, the structure of the chloroplast. Then Julius Robert Redmoyer, they introduced the concept of photosynthesis. Then all of these people, this is formula first time developed by Jean-Baptiste Bosenwald, 6CO2, 12H2O, light energy, then a glucose molecule, a simple carbohydrate. 6 oxygen molecules are released. At the same time, 6 water molecules are also released. Then here, light reactions, dark reactions. Then Robert Hill contributions, all these things. Then this is the formula. Then if we go into the physiology and biochemistry of photosynthesis, what happens? A simplified formula of photosynthesis, 6 carbon dioxide molecules from the atmosphere, water molecules from the soil through the roots. These are converted to inorganic substances are converted to organic. Energy-less compounds are converted to energy-rich compounds. The required energy comes from light. So it is a very simplified formula. But in between these two, there are many biochemical reactions. These biochemical reactions are facilitated in the chlorophyll, through the chlorophyll. The chlorophyll functions as a catalyst. Now again, that photosynthesis reactions take place in 3 stages. 1, radiant energy of sunlight by the chloroplast, absorption of sunlight by the chloroplast. This is first step. Second step, the absorbed energy is converted to ATP and NADPH in the photochemical reactions. So energy present in the sunlight is first converted to ATP and NADPH. In the third stage, the energy present in the ATP and NADP is channeled, converted to an organic molecule, that is glucose. So there are 3 important steps. 1, radiant energy present in the sunlight is absorbed or captured by chloroplast. First step. Second step, the energy present in the chloroplast is converted to ATP and NADH. In the third step, ATP and NADH energy is converted to glucose molecule. So look at here. It is not as simple as we think. The light energy is converted to glucose molecule, we say, but there are so many complicated processes involved. This is the structure of the chloroplast. All of you know. I need not explain. I need not take the time for it. Then chloroplast, important characters. The stroma contains, the chloroplasts are semi-autonomous organelles. They have their own DNA. They can replicate without the involvement of the nucleus. That DNA is double-stranded circular DNA. And chloroplasts contain their own ribosomes, 70S, just like bacteria. The smaller subunit is 30S, the larger subunit is 40S. In addition to that, lipid droplets, starch granules are present. So we have 2 main parts, grana, stroma. Grana, light phase reactions, stroma, dark phase reactions. Then this is very important. This is the structure of grana. This is stroma. This is all stroma. Now this is ATPase, ADP, and inorganic phosphate is converted to ATP. plastocyanin. This is the lumen of the grana of thylakoid, wherein the photolysis of water takes place. Splitting of water molecule into 4 electrons and 1 oxygen molecule. And these protons are used to build the proton motive force. So here chromosome, cytochrome BS1, PS2 with oxygen evolving complex, PS1 with NADP reductase. This is ferredoxin. Remember, this is the structure of 1 granum in the grana thylakoids. Such thylakoids will be made. This is surrounded by stroma. Here the dark phase reactions, here the light phase reactions. Here the light energy is converted to ATP, coupling the ADP and phosphate. For this process, so many molecules are required. For example, plastocyanin, plastoquinone, ferredoxin, cytochrome BF for electron transport system. And look at here, there are 2 types of photosystems, PS2, PS1. This is involved in NADP, reduction to NADPH. This is involved with the formation of ATP molecules. So, photosynthetic pigments. In chloroplasts, photosynthetic pigments are present. Chlorophylls, carotenoids. Chlorophylls A, B, C, D plus related molecules found in prokaryotes called bacteriochlorophyll. There are some bacteria which also perform photosynthesis. There also chlorophyll is present. But that chlorophyll will be known as bacteriochlorophyll, slightly different from the chlorophyll of higher plants. So, in order to differentiate the chlorophyll present in the bacteria, it is known by bacteriochlorophyll. But in higher plants, we have A, B, C, D, including the algae. The A, B are the main, though C, D are there, but main chlorophyll pigments involved in the process of photosynthesis are A and B. Now, look at here. Chlorophyll molecules absorb light in the ranges of blue 435, red 680 wavelengths, and reflect back green. They absorb the blue light, red light. This is around 435. This is around 680. Now look at this. This is chlorophyll A, chlorophyll B. See the difference. Here is methyl group in this particular position, whereas for chlorophyll B, here it is keto group. And this is the head, this is the phytal part, tail part, almost same, only difference lies here. And another important thing, students remember, in the center of the head chlorophyll molecule, there is a metallic ion known as magnesium. The head is a tetrapyrrolic structure. This is a tetrapyrrolic structure. So this chlorophyll molecule includes a hydrophobic tail. Hydrophobic means water fearing. Water doesn't get attached to it. Then this will be embedded in the thylakoid. Then porphyry ring head, a tetrapyrrole ring, circular group of atoms surrounding a central magnesium ion that absorbs. Carotenoids. Carotene, orange, xanthophyll, yellow. So these will be helpful in channeling the light energy to the center of the photosystems. Another is they protect the chlorophylls from photooxidation. This is important point due to excess. So that sometimes the excess light will be falling during the midday. And in order that will be the plants, the green pigments will be protected from the excess sunlight by carotenoid pigments. Otherwise, they get oxidized. That is called photooxidation. So there are two photosystems, photosystem 1, photosystem 2, differences. Again, the same diagram. Photolysis of water. What is the first biochemical reaction in photosynthesis? In fact, that is the splitting of water, photolysis of water. Hill's reaction. Many of you know, during the photosynthesis, during the light phase reactions, oxygen is released. Where does this oxygen come from? Whether is it from water, is it from carbon dioxide? Because in carbon dioxide there is oxygen, in water there is oxygen. The earlier scientists were confused whether it comes from the oxygen or water. Many people thought that it will come from the carbon dioxide. But later on, Robert Hill demonstrated that whatever the oxygen released during the photosynthesis comes only in the light phase reactions, that too from the photolysis of water. So if there is an acceptor, electron acceptor, just like NADP, under natural conditions, NADP functions as electron acceptor. These are called Hill oxidants. Now, we say light is essential for photosynthesis. But light is also a very complicated structure. Look at this. This is electromagnetic radiation. In electromagnetic spectrum, different types of wavelengths of light are there. Long radio, micro, infrared. This is visible to our eye. Ultraviolet, X-rays, gamma rays. Now, if you see the wavelength, as you go in this direction, wavelength decreases, but energy increases. అంటే ఏమిటి? ఈ రేడియో వేవ్స్, మైక్రో, ఐఆర్ వీటి యొక్క వేవ్లెన్స్ ఎక్కువగా ఉంటాయి వేటి కంటే, ఎక్స్రేస్ కంటే, గామా రేస్ కంటే, యూవీ రేస్ కంటే వీటికి తక్కువగా ఉంటాయి. మనం ఇటువైపు పోయినా కొద్దీ ఇవి తక్కువగా ఉంటాయి. జస్ట్ 1 minute. So all these lights are there, but from photosynthesis point of view, only the light of this region is useful. Not these, not these. These are powerful. In fact, these are damaging. These are low wavelength, high wavelength, but low energy, only here. And this is further differentiated into with jar. Blue, red. Wavelength more, red is more, green, blue is less. 400 nanometers to 700 nanometers. అంటే ఒక రకంగా ఇంట్రెస్టింగ్, మన కంటికి ఏదైతే కనబడతాయో, ఏ కలర్స్ అయితే కనబడతాయో అవే రీజియన్లో ప్లాంట్స్ కూడా ఫోటోసింథసిస్ పెర్ఫార్మ్ చేస్తాయి. దట్ ఈస్ అన్ ఇంట్రెస్టింగ్ థింగ్. నేచర్ ఎట్లా ఎవాల్వ్ అయింది అనే దానికి ఇంట్రెస్టింగ్ థింగ్. మన కంటికి ఏ రేంజ్ అయితే లైట్ ఉపయోగపడదో అదే లైట్ ఫోటోసింథసిస్కి ఉపయోగపడదు. సో దిస్ ఇస్ ద వేవ్ లెంగ్త్, ఫ్రీక్వెన్సీ, ఎనర్జీ. గామా రేస్ తక్కువ వేవ్ లెంత్ ఉంటాయి. Now, light phase photochemical reactions. Emerson first demonstrated the existence of two photosystems, photosystem 1, photosystem 2. How he conducted some experiments. He observed that when the light is increased more than 680, there was a fall in the rate of photosynthesis that he called as a red drop. On his name, it is known by Emerson's red drop. But in the second type of experiments, he exposed the chlorophylls, pigments, or the green plants with two types of lights simultaneously. 680 deep red spectrum for red. He exposed the light of both wavelengths. The rate of photosynthesis was found to be higher than some of the red light and far light. This is called Emerson's enhancement effect. Light reactions. When light is available during daytime, mostly during daytime, in grana of chloroplast, light absorption, splitting of water or photolysis of water, release of oxygen, formation of high energy chemical intermediates like ATP, NADPH, that is hydrogen and proton carrier, light absorption, then photosystem structure, architecture. Surrounding molecules are known as antagon complex, reaction center. So it will be like this. So this is reaction center. Okay, mainly consists of chlorophyll A, whereas these surrounding molecules, chlorophyll B, and also carotenoid pigments. This entire structure is called antenna complex. Reaction centers. Photophosphorylation. What is the definition of photophosphorylation? Coupling of adenosine diphosphate and inorganic phosphate known as A to give rise to ATP is known as phosphorylation. We have three types of phosphorylations. Photophosphorylation, substrate level phosphorylation, oxidative phosphorylation. The ATPs are synthesized during the light phase reactions, that is called photophosphorylation. Synthesis of ATP driven by light is known as photophosphorylation. Two types, cyclic, non-cyclic. So this is a cyclic photophosphorylation. Its reaction center, photosystem 2, P680. Photon, light unit of light is known as photon. Then when this photosystem absorbs the light, the chlorophyll molecule becomes excited and loses the electron. This electron again comes back through passing through different electron carriers. So now when the electrons pass through, this proton gradient is established for the synthesis of ATP. So this is cyclic because the electrons that are coming from the photosystem are again coming back. During this process, proton gradient will be established and that will be used for ATP synthesis. So, but at one time people thought that higher plants are cyclic and non-cyclic phosphorylations are different. But now it has been established that these two systems are present. But one dependent on another. They are not independent. Separate ga undavu, ఒకదాని మీద ఒకటి డిపెండ్ అయి ఉంటాయి. దీన్ని ఏమన్నారంటే జెడ్ స్కీమ్ అంటారు. ఇప్పుడు ఇక్కడ చూడండి, దిస్ ఈజ్ వన్ cyclic photophosphorylation, this is non-cyclic photophosphorylation. So whatever the electrons are required for this system comes from this. So one dependent on this is called Z scheme. So Z scheme has been recorded in algae, in cyanobacteria, in higher plants. The non-cyclic cyclic phosphorylation, separate cyclic phosphorylation has been reported in photosynthetic bacteria. So this is differences. So this is an advanced aspect. Now we say in light phase reactions, ATP is first absorbed by chlorophyll pigments. Then that energy is transferred to ATP for the synthesis of ATP. How actually the ATPs are synthesized? This phenomenon, this process has been explained by Peter Midgel through his classical theory known as chemiosmotic theory. Chemiosmotic theory was proposed by Peter Midgel. ATP etla synthesis avutayani. For this he was awarded the Nobel Prize. So this is Peter Midgel. He got the Nobel Award in 1978. So this is, I will show you the diagram. This is actually how when electrons, photons strike the photosystem 2, the splitting of water molecules, the electron transported to photosystem 2, photosystem 1, and whatever the electrons are released, they are used for driving the protons into this. And these protons, when accumulate, more positive charges here, less positive charges here. At one time, these protons try to pass through the ATP synthase molecules. This is ATP synthase. And when the electrons or protons pass through this, ADP and hydrogen phosphates are coupled to form the ATP. So this is the summary of chemiosmotic theory. Actually, it needs more time to explain. So this is what happened. Now, just wait 1 minute. So this is again very important diagram to make you understand. We say all the photosynthetic reactions are broadly divided into light phase reactions, dark phase reactions. What happens in light phase reactions? Light energy is converted to ATP, NADP. This is in total ATP, NADP రెండింటిని కలిపి assimilatory power అంటారు. This is energy carrier, this is hydrogen carrier, proton carrier. So here water is necessary, then protons, oxygen. These are light phase reactions. In dark phase reactions, carbon dioxide, which is in the oxidized state, which is in the inorganic state, which is in the energy less or poor state, is converted to a carbohydrate, which is energy rich, which is an organic molecule, energy rich. So carbon dioxide is not simply converted to a carbohydrate molecule. It is energy less, energy rich. The required energy comes from this. Inorganic molecule is converted to an organic molecule. Carbon dioxide is inorganic, whereas glucose molecule is organic. So here the protons are required. So here In dark phase reactions, carbon dioxide is converted to a glucose molecule. For this, light is not necessary. But the molecules that are synthesized during the light phase reactions are required. So therefore, the dark phase reactions are dependent on light phase reactions. అంటే డార్క్ ఫేజ్ రియాక్షన్స్ కావాల్సిన ప్రోటాన్లు ఎలక్ట్రాన్లు ఎనర్జీ అంతా కూడా లైట్ ఫేజ్ రియాక్షన్స్ నుంచి వస్తాయి. ఈ రియాక్షన్స్ జరగవు అనుకోండి, ఈ రియాక్షన్స్ కూడా జరగవు. ఎందుకంటే ఆల్ దీస్ రియాక్షన్స్, కెల్విన్ సైకిల్ రియాక్షన్స్ ఆర్ డిపెండెంట్ ఆన్ దిస్. So this is what happens. This reverse arrow mark shows that during respiration, glucose molecule will be converted to this ADP phosphate, energy will be released. So of course, I don't want to confuse you. So this is light phase reactions. During daytime, when light is available. During night, when light is not available, light is not required. Light is not required. So this is the relationship between the light phase reaction and dark phase reactions. అంటే మనం రెండింటిని డివైడ్ చేసుకున్నా కానీ వాటికి మధ్య గల సంబంధం ఏమిటి? లైట్ ఫేజ్ రియాక్షన్స్కు లైట్ కావాలి. Light energy is converted to ATP and NADPH. ATP is energy rich molecule. దాంట్లో ఎనర్జీ ఉంది. NADPH లో కూడా ఎనర్జీ ఉంది in the form of protons and electrons. Dark phase reactions are not dependent on light. It doesn't mean that they take place only during the night, during the dark, but not dependent on light. CO2 to glucose. So this is the relationship between these reactions. All both the light phase reactions, dark phase reactions take place in the chloroplast. Okay, light dark phase reaction. Okay, Dr. Tirupati.
Sir.
The remaining part I will take in another class.
Sir, okay, sir. Sir, thank you, sir.
I had to rush through I want to make certain basic aspects to students to understand.
Sir, thank you very much, sir.
Okay.
Kavita Madam Garu.
Tirupati for giving me nice opportunity, for inviting me to deliver the keynote address. I also thank the principals of the 3 important colleges for supporting and encouraging this program. I wish this program will be successful and the students will be immensely benefited by this program. Thank you very much, Dr. Tirupati, and all other students.
Sir, thank you, sir. Thank you very much. Now I would like to invite Srimati E. Josna Madam Garu, lecturer in English, Government Degree College for Women, Jagityal. Please give vote of thanks.
Thank you, sir. So
Malati, where are you working? Gayatri, Jyotsna.
Thank you very much. And now I express my warm gratitude to our chief guest, Honorable Dr.PADALA Garu, for spending
Look, should I stop the sharing?
am I audible, sir?
Yeah.
Please close the share screen, sir. Stop this share screen.
Okay.
Okay. Now, Josna Madam Garu, please proceed. ఇందులో వినబడట్లేదు, ఇక్కడ వచ్చి మాట్లాడారు.
Okay. Okay, am I audible now? Okay,
Yes.
A very good evening to one and all. This is Jyotsna, lecturer in English, and it's my privilege to propose the vote of thanks on behalf of the entire fraternity of GDCW Jagitial. Initially, I congratulate all the team members of GDCW Jagitial. It's my privilege to propose the vote of thanks on behalf of the entire fraternity of GDCW Jagitial. And initially, we congratulate all the organizing committee of this knowledgeable session. On behalf of our institution, I extend our sincere gratitude to our renowned keynote speaker, Professor S. Reddy Garu, Department of Botany and Microbiology, KU Warangal, for accepting our invitation and briefing us with the theme of photosynthesis. Thank you, sir. Thank you for gracing this occasion. And also, I express my warm gratitude to our chief guest, Honorable Dr. K. Surendra Reddy Garu, for sparing his precious time for us and gracing this occasion. And we wholeheartedly thankful our honorable and remarkable principal, GDCW Jagitial, Dr. Y. Satyanarayana Garu, for his unceasing support and encouragement to make the wonders. And also, I heartfully extend my sincere thanks to the renowned principal, Honorable Dr. K. Ramakrishna Garu, for gracing this occasion. And we also extend my sincere thanks to the honorable principal, RBVRR Hyderabad, Dr. J. Achyuta Devi Garu, for her continuous efforts and encouragement to make all this possible. And also I express my heartfelt gratitude to our honorable